All right, I'm going to talk about active transport with you next. And active transport is going to be very similar to our passive transport. Some features are going to be flip-flopped, and it's pretty easy to remember because of its name, active transport. Let me get myself kind of hidden over here. All right. So active, when you are active, you are using energy, right? When you're outside running around, you're playing whatever sports, you are, I don't know, actively learning, you're using energy to perform whatever it is. So active transport for our cell membranes are going to use energy to move um, specific molecules against their concentration gradient. So we're going to go back to my little analogy on the hill here, right? So this time we're going to have an area of um, low concentration down here because we're at the bottom of the hill, low, right? And we're going to move substances from low concentration to high concentration. So here's my boulder. I need to move it up the hill. I need to use energy to do that. And I'm going from an area of low concentration to an area of high concentration up here. So it's going against the gradient. And that energy that helps move it is known as ATP. Adenosine triphosphate. We'll eventually talk about that and how it's usable energy currency for the cell. But just generally know that ATP helps in active transport. And there are going to be a couple different types of active transport. There will be pumps. So these are our protein pumps, much like what we saw in the previous one, but we can see there are specific shapes for each molecule for it to go in and out. We can also see that feature of how there has to be ATP. Here it comes. These yellow molecules will attach so that it can change its conformation and allow things to go in and out of the cell. And then there's a second type known as bulk transportation. And this is where those vesicles, those our lipid bilayer vesicles will surround themselves around whatever the substance is, and they'll move them, send them out of the cell, or maybe they're moving them and sending them into the cell, but they're using these big vesicles to do it. Um, So the cell membrane pumps that I was talking about are going to be based off of membrane potential, but it has to deal with unequal voltage distribution. So that means we could have some charged molecules, we have some ion pumps that will help with that. And the most common example, which we saw in the previous one, is known as the sodium potassium pump. Sodium, the element is Na, right? Sodium is not good for our cells because that leads to dehydration. And the potassium is good for our cells, right? Think bananas. We like that to help keep our cells healthy. So that's the sodium potassium pump, which will take out 3 sodium ions and then bring in 2 potassium ions every single time. And again, ATP is going to be that molecule of energy that is going to help pump those molecules across the concentration gradient. So I'm just going to go back, if I can, quickly and talk about, here's our sodium potassium pump again. Here's our sodium molecules. Potassium is green if you are not colorblind. Here's the inside of the cell, right? We should have a higher concentration of potassium in the cell because that's good, and there will be a lower concentration of potassium outside of the cell. So anytime there is potassium, it hooks up to one of those carbohydrates on the outside of the cell and says, hey, I've got potassium. We want it. Let's pump it against the concentration gradient and bring it on into the cell, right? So it'll hook up and link up into its specific areas, its active site. Energy will come and change the conformation of that protein so that it can enter into the cell. Then we've got our yellow or our smaller little sodium molecules where there's going to be a higher concentration of sodium outside of the cell because that's bad, right? Lower concentration on the inside. We want to get rid of those sodium ions. And so 3 of those molecules will come and attach. ATP will again come change the shape of the protein and they'll be kicked out. And then just like a personal note on this one of my really, really close friends, they have a little boy who has an issue with this sodium potassium pump. So if this isn't doing what it's supposed to, it can lead to several different factors. One of them is something known as cystic fibrosis, which we're going to be doing a group case study on that. But if you can't pump sodium out of the cell, right, that's going to accumulate for that individual, and that can be very dangerous for all sorts of reasons, like, developmental reasons. So whether it's just growth or if it's related to the brain or physiological changes. Um, One way of identifying that cystic fibrosis is based on how salty your skin is. So if you know anyone with that, you may know like that's just one thing they do. They'll do a sweat test to identify it. But my close friends, they have a little boy who's about a year old now, but he has an issue with his sodium potassium pump, which led to a lot of seizures and things like that. And they really couldn't figure it out what exactly was happening. But again, it was an accumulation of too much sodium staying inside of the cell and not enough potassium coming in, and that led to several seizures. And he's still growing and developing, but he's much further behind than any other 1-year old there is. So they have a lot of work to go. But he takes medication that helps um, with this transportation process. Okay, sorry, that was my side note. Bulk transport is the other version of active transport. Maybe I can pull myself down here. Yeah, whatever. Okay. So bulk transport can be broken down into a couple categories: exocytosis and endocytosis. So if you take the prefix exo, that means exit, endo is inside, and then cytosis is referring to the movement within the cell. So outside of the cell, inside of the cell. So vesicles or membrane-bound vesicles are going to contain whatever those molecules are, whether they're good or bad. They'll have them surrounded here. And then that vesicle is going to fuse with the plasma membrane, and then it'll either bring those molecules into the cell, if we're talking about endocytosis, or it will push them out of the cell, which would be called exocytosis. So this example right here, we see our vesicle forming and coming into the cell. So that's endocytosis, good stuff in, bad stuff coming out. So it's going to fuse with the membrane and push it out. Again, it's going to go against the concentration gradient. In order to move these huge vesicles with all these molecules, there's going to be some ATP that hooks up and then helps, push it to the direction that it needs to go. So that's bulk transportation. There are also a couple of versions of endocytosis that we should be aware of. And these are kind of cool. Phagocytosis is talking about food specifically and how that gets brought into the cell. So there are specific organisms that go through and use their vacuoles to feed. And they will go ahead and use phagocytosis by engulfing something and then bringing it in to be broken down by the lysosomes and digested. Penocytosis is bringing in fluids. So that again is endocytosis. Fluids are coming in, but it's going to be that vesicle for, various fluids besides water, which can freely flow across the cell. And then receptor-mediated endocytosis is this example over here on the side. So we see these green circular pieces. Those are the receptors. So those are going to communicate, hey, there's something that is attaching over here. Is it good? Is it bad? And this is going to be something more like kind of like the medication stuff, like that will be able to enter into the cell. So these little receptor proteins will activate. Once that molecule attaches, then the vesicle fuses around it and brings it on into the cell. So those are our versions of endocytosis. The end.